So we've talked about most of the organs of the alimentary tract. We've talked about the mouth, the oral food processing, that first bite, the chewing, the springiness of cheese. Remember, we showed the cheese curds to illustrate the importance of food oral processing when we talk about sensory properties. We also talked about different enzymes that are produced. I'm just going to turn this off. We also talked about different enzymes that are produced in the mouth. Salivary amylase. Remember, salivary amylase cannot break down carbohydrates. Cannot break down carbohydrates into monosaccharides, so we require the brush border enzymes. Okay, how do I get rid of you? Go away. Ugh. Oh, I got to stop something. Yes, it recorded. Sorry. I recorded my screen so we have a good video of the conversation with Nick. Come on. What are we doing? Okay, so we need to swap displays. There we go. Go back, go back. Okay. So in the mouth, we've got the oral food processing. We've got the reduction of particle size. To form that bolus, we not only need the reduction in particle size, we need the increase of lubicity. So we have to produce salivary amylase. So we have particle size reduction and absorption of water into that food, which allows you to form the bolus and swallow it. Once it reaches the stomach, we have a change in pH. That change in pH kills bugs, which again, we talked about it being good when we talk about pathogens, but bad when we talk about probiotics. We also see a denaturation of proteins, so those proteins begin to unfold. Why is that important? It slows down gastric emptying. If we slow down gastric emptying, we change the interaction of the body with the food. That means we change the level of satiety that's induced. So when we see the transition, so we have that change in the stomach, the acidification, the denaturation. It then goes into the small intestine. The small intestine, weird. No. The small intestine is where the enzymes interact with the food. Pancreatic amylase. We have pepsins, we have trypsin, we have chymotrypsin. We have the lipases in the stomach. We also have the major part of absorption for those macronutrients. The food then moves to the large intestine, and we're going to talk about the large intestine again. We talked a little bit about it last day, but I want to slow down and talk about it again. So when we talk about the large intestine, we're talking mainly about water absorption and further degradation of the non-digestible constituents of foods. So these are things like your fibers, hemicellulose, pectin, all of these different ingredients that are naturally found in plant materials, but are also extracted and used in the food industry. So things like carrageenan being used in chocolate milk to stabilize the suspension of the chocolate particles, or alginates being used in molecular gastronomy. So the colon houses the microbiome that's able then to interact with foods, to metabolize them, and by metabolizing them, they produce different constituents or metabolites that can then be absorbed by the body and then trigger other effects. So when we talk about the microbial population, if you have a healthy microbiome, there is a lot of synergisms between the host and the microbiome. There's the production of short-chain fatty acids. And we talked about the incorporation of short-chain fatty acids into our food. Remember when they're cleaved off the glycerol, things like things like C2, C3, and C4, so 4 carbons and less for the aliphatic chain length. Those can be used as an energy source by the epithelial cells. So we already talked about that when a short-chain fatty acid is present in foods. With the microbiome, they are able to convert non-glycemic carbohydrates, things we can't digest, and they can produce short-chain fatty acids. And those short-chain fatty acids are extremely important in assessing gut health. Now, again, so non-glycemic carbohydrates can be fermented and can produce short-chain fatty acids. We already talked about binding bile. Why is that important? If we bind bile and it is excreted, what do we have to use to produce more bile in the liver? Someone? Yes. Cholesterol. So by binding bile, or by eating a high-fiber diet and binding bile, which is then excreted in the feces, we have to rely on our systemic circulation and the cholesterol that's in our body to reform additional bile. So it's an excellent way of reducing cholesterol naturally. Now, when you're born, from a microbiome perspective, you are sterile. You have no colonization of your colon yet. This is determined very, very early in life. If you're bottle fed, or if you're breastfed, if you're born by cesarean section, or if you're born vaginally, it begins to influence what microbiome is established in your body. If you've been born in a hospital or at home, if you've had antibiotic use, all of these things affect the population. Now, when we talk about the population of microorganisms, and again, this is so difficult to kind of comprehend. If you talk about every single person in here having 300-400 different species of bacteria, there's been reports that some people have up to 1,000 species of bacteria that have colonized their gut. So think about it. We have these beneficial bacteria that are all producing short-chain fatty acids. Those short-chain fatty acids are being absorbed. They're influencing gut health, colon health. They're influencing, they get picked up by the liver. So all of you have, let's say, 300 different species of bacteria. The ratio, or the relative proportions of those bacteria are determinants in health. And we know a very small amount about how they influence health. So again, the population distribution of the bacteria for each one of you is drastically different. So when we make food choices based on what those microorganisms can digest and ferment and produce, every one of you will have a different response to food. So one of you could consume a food that's benign, but if you have an exotic bacteria that's producing a metabolite that is a carcinogen for us, that can be highly problematic. And an example of this is if you get a C. difficile infection. Extremely hard to treat without wiping out your entire gut microflora. So how do you begin to treat chronic exposure to bacteria that are carcinogenic or that have other detrimental effects? It becomes very, very challenging. Why is that not going now? My computer's thinking. There we go. So again, that population when you're born is non-existent. Very quickly we establish a gut microflora within a few weeks, months after birth. And that population evolves as you age. So in healthy adults, our bacteriocytes, I hate saying these. I hate the next 2 weeks, the microbiology. It's like pain for me. And the firmicutes. The ratio of those change through life. And they are markers for health. The other really cool thing is that they've shown that that population or that ratio changes as you lose weight. So if we look at that study, so the two data points are from this Nature paper. So the first graph at the top shows what happens if you take a fat-restricted diet, or, so the fat-restricted diet is in the bottom graph, the red dots, and if you have a carbohydrate-restricted diet. So these people that have taken this study have lost weight. So if in this study you lost weight on either the carbohydrate-rich diet or the fat-rich diet, if you lost 2% of your body fat, you were included in the study, and if for fat, you had to lose 6%. But look, you can see that your population ratio between your bacteriocytes and your firmicutes change as you lose weight. So as you're dieting, as you're restricting the amount of calories, that population of your gut responds. So there's this amazing initiative being run out of the University of California, and I'm hoping to get them to Skype into class, but it's called the American Gut Project. Has anyone ever heard of the American Gut Project? Nobody? So what they're trying to do is understand how the population's gut microflora varies. So what you can do is you can go online and just search American Gut Project, donate $100 to them, and they'll send you a swab. And you take that swab and you take a fecal sample and you send it back to them. And they will send you what your ratio of your bactericides and firmicutes are, but they'll also tell you if you have any bacterial populations that are really high or really low in comparison. So we talked about Emma Allen Van Crow, who's a gut microbiologist here. She did it, and she found that she had a bacterial species that was 10-fold higher than the rest of the population that plays a major role in colon cancer. So it becomes very important. So with this, I'm doing the American Gut Project as part of my diet. So I've done the first one, the first fecal swab, and I'm going to do the next one in November because it takes about a month for them to analyze the results. So we'll see how my population of gut microbacteria change in response to weight loss. And we'll talk about how that affects. So the ecology of the microbiome is dependent on how overweight you are. The other thing is, and again, we are still at the infancy of this science. There is so much material and interactions that we have no idea of the ramifications. But when you get into dysbiosis, and dysbiosis is when you have a microbiome that has shifted or been influenced, and this is a maladaptation, typically leading to some kind of impairment. We know that the gut microbiome is able to interact with the PYY hormones, right? If it's able to interact with the PYY hormone and influence satiety, could this be tied to obesity? So if all of a sudden these microbiomes are interacting with PYY, could they regulate our appetite? When we get into dysbiosis, there's all kinds of diseases now that have been implicated in dysbiosis. Things like autism. There's a conversation around. Things like mood and behavior are affected by the metabolites that the bugs produce. But it's more than that. It can affect things like colon cancers. It can affect other inflammatory diseases, irritable bowel syndrome, Crohn's disease. All of these implications are being studied when we talk about the human microbiome. So there are very important relationships between the human body and the gut microbiome. Again, we don't understand all of them. We know that short-chain fatty acids, again, the metabolites from non-digestible carbohydrate digestion, are extremely, extremely important at determining how healthful you are. So those short-chain fatty acids, again, can be taken up by the cells and reducing the incidence or the likelihood of developing cancers. We also know that those short-chain fatty acids are metabolized, or they go in through gluconeogenesis through the liver. So they play a very, very important role in health. And again, the healthy adult can have as many as 1,000 species. There's really 300 or 400 that are really relevant. But again, when you're talking about a population of 0.1% of your microbiome, can have a massive impact on your health. We're starting to understand. So again, there's a lot of important interactions between the microbiome and the human body. And again, this is staggering. When we talk about the number of genes, and genes code things like enzymes. So if you have 30 times more genes, you have 30 times more enzymes typically. Now, if we have 300,000 genes that are shared by greater than 50% of the population, that means there's a lot of similarities between the microbiome for each of us. But there's a lot of variability as well. So again, we talked about that humans can be classified based on different aspects of how they've lived their life. If they've been breastfed or infant formula fed, will change your microbiome into adulthood. So again, when we talk about ramifications of the microbiome being able to influence health, this is important. So is there a difference between formula feeding an infant and breastfeeding an infant? There's all kinds of studies that show that those two things are very important in determinants of whether or not you become obese later in life. Is that related to then your microbiome? We don't know. And again, the other classes. So whether or not you're educated or not changes the microbiome. And again, this is remarkable. The exposure to foods differs between if you're from a low socioeconomic status or a high socioeconomic status. So the foods you consume differ. That then changes the microbiome that you have in your colon. And when you think about this evolutionary adaptation, which I keep coming back to in this course, but take things like lettuce and onions. When we talk about some of the non-glycemic carbohydrates that we can't digest, it's remarkable to think that we can convert them, the microbiomes can convert them to something that's usable by the human body for energy, so the short-chain fatty acids. So just to give you an example, 92% of this class will have a microbiota that is able to break down xyloglycans. Xyloglycans are found in all kinds of vegetables and fruits. It's just a non-digestible carbohydrate. So from an evolutionary adaptation, the energy that you get from those is important. Especially when you're talking about if you're a hunter-gatherer and all of a sudden the majority of your diet is coming from plant-based, low-caloric foods, the advantage of being able to derive calories from non-digestible constituents is extremely important from an evolutionary standpoint. Remember, for most of human evolution, we have been spending 90% of our time hunting and gathering. So by increasing the caloric density of a food, we can spend less time foraging for food. But now it has consequences in health, because those metabolites are absorbed and then they interact with different processes in the human body. So things like cardiovascular disease. So there's the TMAO. So TMAO is a marker of how much stress your heart is under. We know that phosphatidylcholine, which is lecithin, which is found in eggs, which is found in most plant materials, when that is metabolized by gut microbiome, those metabolites negatively impact coronary vascular disease. We have indications that that it has a role in inflammatory bowel disease. And again, phosphatidylcholine not only is commonly found in many, many foods, it's an extremely important food additive. It's a great emulsifier, which we'll talk about later. So Emma Allen Van Croo is a heavyweight when it comes to the gut microbiome. She is one of the top researchers internationally. So her research really focuses on dysbiosis of the human microbiome. So what happens when your microbiome is altered in a negative way? What can happen? So talking about colon cancer, again, those short-chain fatty acids and interacting with the epithelial cells. And her research is remarkable in that she studies a very different aspect of probiotics. So when we talk about probiotics, we typically consume them orally, right? What's the challenge to that? They have to survive the upper GI tract, which is challenging. You've got low pH, which is not desirable for microorganisms. You've got bile, which is not desirable for microorganisms. So how do you bypass that? The other end. So instead of taking it orally, by taking it rectally, you can actually ensure that that bacteria culture that you're putting in there has survived and is able to colonize it. So there's this huge fad right now, especially in the United States, to do fecal transplants. So you take a healthy donor's fecal material, and you take an unhealthy recipient, and you give them a little enema. And again, Emma Allen Van Croo is a world expert in exactly that. Like, her lab is designed to understand how how you can take that microbiome out of a healthy individual and study it and be able to examine it because it's extremely challenging. Now, when we talk about the fact that our microbiome has 30 times the number of genes than the human microbiome, we have to understand that there's different enzymes that are produced by the microbiome. So we don't produce azoreductases, nitroreductases. We don't produce any of those. So when we have a food that has a chemical bond that these enzymes are able to break apart, we then have to be concerned with the metabolites that have been produced, right? So we have healthy food A compound. It's got a specific bond that one of these enzymes can reduce, and all of a sudden there's either a new compound or that molecule has been split. So now, equally as important to the toxicity of that original molecule, we need to understand the toxicity of either the new molecules or the new molecule, depending if it's been cleaved into two molecules or not. And if we don't study that, if we don't understand how these artificial dyes, how these artificial flavors interact with your microbiome, we won't know if the metabolites they produce are carcinogenic. So we could be constantly, chronically exposed to metabolites of GRAS status compounds, dyes, pigments, flavors. They could be metabolized into very, very toxic molecules that over a lifetime have implications in things like cancers and heart disease and diabetes. So this whole area of the human microbiome is extremely important. And we're going to talk about one reaction and one enzyme. And this is with our azodyes. Azodyes are found in all kinds of foods. Things like Skittles have dyes, artificial dyes. So we have things like tartrazine, sunset yellow, allure red. These are all pigments that are found widely in the confectionery industry, in things like toothpaste, in deodorant, in shampoo, in conditioner. We are chronically exposed to artificial dyes. So when you talk about the enzyme, the azoreductase, being able to interact with a molecule or a pigment, so we have methyl red here. If we're able to cleave this double bond here, we get two different molecules. Now, this one molecule on this side, NN-dimethyl-P, what's the last part of it? Dimethyl-P phenyl, oh jeepers, phenylene diamine, is a potent carcinogen. So we take methyl red, which is found in cosmetics, which is found in all kinds of products, not in foods, and we cleave that double bond. All of a sudden, a bacterial population can metabolize that, produce N-dimethylidine, and now what effects does it have? We know it's a carcinogen, so if you're eating that every day and you're getting small amounts of this NNDP, all of a sudden we have a huge issue with safety of that ingredient. And to be honest, if you look at the world, we are extremely, Canada specifically, is extremely far behind in our restriction of artificial colors. If you look at the European Union, none of these E-number dyes are in our foods. In Canada, we have a lot of artificial dyes that are still allowable, and there's a lot of evidence to suggest that there's detrimental effects to them. And this is an amazing collaboration here at the University of Guelph. So Emma Allen Van Crow is an expert in the gut microflora. Giselle Lapointe is in the Department of Food Science. She is an expert in probiotics, specifically looking at dairy, so yogurts, cheeses, probiotics that may be taken in supplement form. She is, again, one of the world experts in this area. And Dr. Giuseppe, from when I was a student here, he actually taught me a course, and at the time he was understanding what or how metabolites of bacteria can affect things like breast cancer. So he's again an expert in understanding these azoreductases and sulfur reductases. So this consortium of professors here at Guelph has just finished a really cool study understanding how colors that are commonly found in foods could play a role in health. So what they've done is they've looked at the change in the short-chain fatty acids and the metabolites that are produced when you chronically expose bacteria to a different dye. And this research will be published pretty soon. Come on, there we go. So, a quick recap. Food enters the mouth, We orally process it, we chew it, we masticate it, we're mixing it constantly with saliva, we're lubricating that food, we're reducing the particle size, we form a bolus, we can swallow it. While it's in the mouth, we have salivary amylase, lingual lipase. Those begin to break down the foods. It then moves into the stomach, pH drops, bacteria die, proteins denature, lipase is released, protease is released. It then travels into the intestines, bile is then incorporated, pancreatic amylase, pancreatic pepsin, chymotrypsin, trypsin are released. They begin to degrade those macromolecules into smaller and smaller groups until we have monosaccharides, amino acids, fatty acids, and two glycerol or two monoglycerides. Those are able to be absorbed. They're either transported via the portal vein to the liver. So if it's water soluble, they go to the liver, they're first pass metabolism. Fats get picked up by the lymphatic system and they go through systemic circulation. The colon has a lot of water absorption, has bile reabsorption, but also the gut microflora. That gut microflora is able to digest foods that we are unable to. The metabolites from that bacteria are then absorbed or interact with our hormone receptors in our gut and can influence dysbiosis and disease. The food is then excreted. So glucose we need to then assimilate. We need to put it back together. So if we have excess glucose, it's converted either to glycogen or to fat. Amino acids go to the liver for protein synthesis. Again, we talk about things like your VLDL. We have to detoxify the ammonium and the urea. And we have chylomicrons. The chylomicrons, again, are from the absorption of your lipids, which then go from your intestinal cells to the lymphatic system, which then can undergo oxidation once they reach the sites. So again, this continuum of food, and what I really want you guys with this class to understand, is that food is extremely complex. But what's even more complex is how our human body interacts with that food, in determining satiety, in determining hunger. So all these hormones have this cyclic nature that drives feeding. So whenever we can modify or manipulate how our body interacts with that food, we need to understand the biological consequences of chronic exposure to processed foods. We do not understand it currently. We don't know what happens when you're, well, we know a little bit about what happens when you're chronically exposed to, let's say, high glycemic foods. But think about everything else we change to the food industry. How we process foods, combinations of ingredients or macromolecules that we would have never seen in an evolutionary path. These small changes, things like cooking, will influence the healthfulness of you when you're in your 40s and your 50s. So we have to be able to understand, and the whole purpose of this course is going to be hopefully to make you realize that when you talk about nutrition, when you talk about agriculture, and when you talk about food science, if you talk about them in an isolated silo without thinking of the other ramifications, we are going to create and have created massive problems with the healthcare system. We have 230 million people in North America that are undergoing treatment for metabolic syndrome. Think about the costs that are associated with that. You know, what you are doing in this class is extremely, extremely, extremely important. And with that, I'm going to give you a little teaser for tomorrow's lecture. Or not tomorrow, sorry, next week's. So if anyone has been following the news, which apparently not many of you do, there has just been a massive recall. So next week, we're going to start talking about safety of foods and how do we produce a food that we can put on a shelf for 6 months and not kill you. So that's next week's lecture. So with that, I will see you next week.